Hey, this is Mrs. Hunt, and this is Module 3 going through today. Hope you're having a great day today, and you're going to have a good week. All right, first of all, I posted a reflection piece, and I recorded it in here, and it just kind of goes over the things in the past. I also hit on this document up here, Roland, due dates. So I'm going to keep that posted up at the top for due dates. Believe it or not, I mess up on due dates. It's hard to kind of think that seventh period comes first with my Monday, Tuesday, and sixth period comes last with my Wednesday, Thursday. So as I'm getting through stuff and you don't understand something, please ask me to clarify. Some of you guys have done a good job in catching me when I've messed up. So please make sure my seventh period kids, your work should be due. If you're a Monday kid, it's Monday. If you're a Tuesday kid, it's Tuesday. If you're virtual seventh period, it's Tuesday. If you are in my sixth period, you're Wednesday, Thursday for me. If you have Wednesday, your work is due Wednesday. If you're Thursday, your work is due Thursday. And if you're a virtual sixth period, your work is due Thursday. Please get your work in on time. All right, let's go through some notes real quick. We're going to go into chapter 6, and we're going to take a tour of the cell. Boy, I get excited when I look at the cell, and I think about how complex it is and how it's put together. It's pretty exciting to think about how we're made in such tiny detail, and it's pretty neat to think about the intricacies of that cell, and then how that cell responds to stimuli, how it responds to oxygen coming in, how it responds to, like we talked about 2 weeks ago, iron coming in, iodine coming in. All these kind of things are pretty neat to think about. Okay, so we're going to go through the cell real quick, and hopefully you remember a lot of stuff that you did in honors biology with me. Much of the chapter is considered required requisite knowledge. So focus on anything new. And as your teacher of the old, I've tried to say, now that's something I didn't teach you. So I'm going to try to make sure I'm going through this. This was a good quote from Emily Dickinson. Faith is a fine invention when gentlemen can see, but microscopes are prudent in an emergency. So I thought that was pretty good. All right, your Latin stuff, sight means cell, and ology means the study of. You should look at observations of several different microscopes, which we're going to do in the classroom. My virtual kids, I'm going to link some pictures to some slides for you so you can see what they look like underneath the microscope. So the beginning of this is going to be talking about microscopes, so we can see why we need to see something microscopic. And it talks about the size, so you can see what the average human height is. Some of our nerve cells are super long, and muscle cells. Sometimes whenever they're doing a cell transplant or a nerve transplant, and they'll take out a nerve of your leg and transplant it to another part of the body. I was always amazed, on movies and TV shows, they would always cut out a nerve that would be like this long. And I would think, that's one cell. But now we're starting to learn that nerve cells and muscle cells can actually be really long. You can see the size of a chicken egg, a frog egg. And then we go to using a light microscope to be able to see most plants and animals, the nucleus and bacteria. Can't really say 100% that I've seen a mitochondrion using my light microscopes. This symbol over here is micrometer. So we have millimeter, centimeter, and micrometer. So we're getting really small. And we go to nanometer. We're going to look at the electron microscope also. At the University of Tennessee in Knoxville, we had an electron microscope. I was not allowed to touch it, but we had one, which was pretty exciting because those are really, really expensive to look at. All right, light microscopes uses light. They're relatively inexpensive. We can examine things. We ran through this in class last week, and your interactive for microscopes should help you. We looked at the eyepiece, or the ocular lens, and the pillar. We looked at the nose, and the objective lenses, the arm, the stage. Remember, we ran through the diaphragm down here, our light source, and then the base. And we used the coarse adjustment and the fine adjustment, or coarse adjustment and fine focus. Here you can see the benefits of stains. We love to use methylene blue or bromenthal blue to stain the cells, so you can see it better under a stain. Here's another technique of using contrast, so you can see the results are different. Resolution is the ability to detect discrete points and separate from each other. As you increase magnification, your resolution is going to decrease. And light microscopes are good for 100 to 1,000 times magnification. They do have their limits, and that's true. So the structures, some of the tiny organelles are beyond what you're going to be able to see. You'll need to make other ways to observe them. Fluorescence, using dyes to make parts of cells glow, is a really cool thing. I'm hoping that we get to do a lab like that where we're going to look at genes being passed on and we'll make some things glow. So that would be a really cool lab. So there's different ways that we can be able to, add more visual into the cell. And then we get into looking at electron microscopes. So these were invented in 1939. They weren't used very much. This is a transmission electron microscope. And then this is a scanning electron microscope. I mean, look at the crazy detail in all of these pictures. The advantage, you can see things better. They can go up to 50,000 times or higher if possible. Can even get down to the atomic level, firing electrons through something. You do need a vacuum. The specimens must stop the electrons. Super high cost to be able to do this. So both of these are going to send electrons through a thinly sliced specimen. And it gives the magnification of the interior. So basically the electrons are firing and they bounce off of something or they bounce through it. And whatever the shadow is what you're going to see is the contrast from that. The specimen does have to be dead to be able to see this. So you get excellent views of the surface, and we can see cell fractions, chromatography, and then electrophoresis. We're hopefully going to do some gel electrophoresis where we look at proteins and DNA and do that in the classroom. Here's another one that they're going to use where they're going to blend up the tissues in the cell, basically releasing the individual cells to be able to come through here. And they're going to use a centrifuge to spin it out and hopefully get the ribosomes to come out. Chromatography technique is another one where you're looking at paper, glass, and columns on a thin layer. And then the electrophoresis is using the movement of electrical field. So it produces like a pole. So you attach a battery to one side and it makes a positive charge and it causes the DNA or proteins to move across. History of the cell. We want to say thank you, Robert Hooke, for discovering the cells in cork. I wish that you hadn't named them after a prison cell, but you did. So that's what we got cell from. So in Latin, it means tiny rooms, and he was thinking it looks like a prison. So that's where we got them. We got Robert Brown, who discovered the nucleus in 1833. Matthias Schleiden and Theodor Schwann also made great discoveries. And Matthias Schleiden, Theodor Schwann, and Robert Hooke basically give us the premise for our cell theory. If you remember back in the day, I made you guys listen to an awesome song about the cell theory. But basically, you should know all living matter is composed of one or more cells. The cell is the structure and function of all life, and all cells come from pre-existing cells. So we get into some questions there with evolution. Types of cells, we got prokaryotic and eukaryotic. If you remember, I would say pro, no nucleus. But remember, pro doesn't mean no. If you're pro something, you're for it. So pay attention to that. Prokaryotic cells lack a nucleus and membrane-bound organelles, and eukaryotic cells have a nucleus and membrane-bound organelles. So evolutionary, we would think prokaryotic came first, and then eukaryotic came later. Cytosol is kind of a newer word. This is the aqueous component of the cytoplasm. We always talked about cytoplasm as kind of like a jello kind of structure, but cytosol is more of the liquid part of that cytoplasm. So that's a different word for you. Ribosomes. We're going to get into a little bit more detail about how ribosomes are different. So here's the prokaryotic cell. You can see it's got a cell membrane, a cell wall possibly, and then inside is just going to be loose DNA and some cytosol and then a ribosome. And then we have the complexity of a eukaryotic cell. Okay, here we go again, looking at a bacterial cell. This is bacilli shape. It's that rod kind of shape or pill shape looking. This has got flagella hanging off the side for transportation and movement. And it's got cilia also for movement. We've got a cell wall, plasma membrane, ribosomes. The nucleoid is just where your genetic information is, the chromosomes and the DNA. All right, again, looking at the size difference. Look how small a prokaryotic cell is compared to the eukaryotic cell. So why are cells so small? Make sure you watch the video on agar cubes in here. It's a good demonstration. We did it in honors biology, and for the kids in the classroom, I'm going to run through another demonstration to just show you guys. But again, it reinforces that small is better. When we think about diffusion, remember movement of particles from high concentration to low. As I drink liquid or water, I want it to diffuse throughout my whole body. If everything is small, it's easy to move very quickly throughout the cell and then to the next and then to the next. And smaller is more efficient. So we say the cell volume to the surface area ratio favors a smaller size. The nucleus and cytoplasm consideration, and then metabolic requirements. So here we have one tiny little cell, and we'll see that it's got a total surface area of 6. The volume is a 1. So the surface to volume ratio is 6. This is a much bigger number. So the outside is bigger than the inside. When we look at a bigger cube, we're saying that the ratio is really low. So this guy is really hard for things to move through. So oxygen, CO2 are going to have a hard time moving through. It's going to take a lot longer to get all the way through the cell and then pass it on to another one. All right, basic cell organization. Remember, the cell membrane is on the outside, the nucleus, cytoplasm, and organelles. And we're going to hit all of those. The animal cell, here we go. So this is an animal cell, and I want you to take time to go through all these organelles. I'm not going to spend time reading them to you all right now. But notice the orientation. So from the nucleus, when you're coming out, we have that endoplasmic reticulum. And it's very close to the Golgi and Golgi apparatus, Golgi body, Golgi complex, however you call it. And then we have our mitochondria as we move out of the cell. So make sure you're paying attention to all that. The plant cell, similar, but we have that chloroplast we're adding in here and that very big vacuole. Remember, this fills up with water and creates turgor pressure, be able to make that cell plump. If you've ever watered a wilted plant before, you basically are putting water into those vacuoles, and as the vacuole expands, the cell expands and plumps up. All right, the membrane separates the cell from the environment, and the boundary layer for regulating the movement. Remember, this is semi-permeable, which means some stuff comes in, not everything. Cytoplasm or cytosol is listed here, the cell substance between the cell membrane and the nucleus. And organelles mean tiny organs or all the little parts inside, the subunits in that cell. Okay, so you got to be able to identify the major organelles, give their structure and function. So continue going through here as we're going to hit all the different organelles, and I'm not going to read all these over to you. In the book, they do some great jobs of looking at some big visuals of the outside of the cell and the inside of the cell. So make sure you guys are going through there. I'm just kind of flipping through to see if there was anything else I needed to talk about. So we're going through all those organelles. This is going to be something different. We're talking about the microtubules, and we start talking about cilia and flagella. This is something newer. Remember, cilia and flagella aid in locomotion, aid in movement. Your cells at this level are not thinking or cognizant, but they are just responding to the environment and moving. The dynin are the motor proteins that connect to the tubes. I want you to watch. There's a video I've given you on swimming sperm of ferns. It's pretty amazing to think about a plant having mobile or modal sperm. So you can be able to watch that video and see that. And then this is what's connected to the cell that aids in that movement. So these appendages are pretty concentrated types of microtubules. They use a contractile protein. They use ATP, which is energy. So hopefully you remember that. All right. Okay, so I'm going to stop there and